We'll make a start. I'd like to kick off by celebrating and acknowledging the traditional owners and custodians of the land upon which we meet today, the Wurundjeri people of the Kulin Nation. Pay my respects to their elders past and present and embrace their continued connection to this place. And I'd also like to extend that respect to elders from other communities that might be from where people are joining us online today. Now, it's my absolute pleasure to introduce today's Institute seminar. Kelin Zhao will be delivering her PhD completion seminar. Kelin was supervised by myself and Verena Wimmer from the Centre for Dynamic Imaging. And when I say supervisors, there were definitely times where it felt like Kelin was supervising us. I'm sure Verena would attest to that. She'll share with you a fascinating story about a thymus, an organ that is central to immunity. And even now, 6 decades after Jacques Miller discovered the immunological function of the thymus, it still surprises us with weird and wonderful insights, and Kelin will share with you some of those today. And I think this project showcases some of the qualities that Kelin brings that distinguish her for a future as an excellent researcher. Number one, she's technically gifted and bold. So Kelin saw the potential for new imaging modalities to shed new light on immune function, and she's proven remarkably skilled at experimental design, execution, and analysis, and together in collaboration with the Centre for Dynamic Imaging, ably led by Kelly Rogers, has created new methods that have powered the discoveries she'll share with you today. 2, Kelin is extremely hardworking. This is an often overlooked but essential ingredient in success. And everyone who knows Kelin is familiar with her constant presence in the lab and also at her desk, powered by all sorts of unhealthy snacks. So if you're ever at a low point, I learned that you go to Kelin's desk and there's always more than enough stuff to buoy your spirits. She's on that high right now, I'm sure. Number 3, she has grit and is very determined, which is sort of a nice way of saying she's very stubborn. Another useful trait for a scientist. As you'll see, Kelin made a striking finding early in her PhD studies, and like many surprising discoveries, I met it with mild skepticism at first, and this was like waving a red flag at a bull for Kelin. Like, she was enraged in the best possible way and spent the next few years trying to prove me wrong, which I'm delighted about. Great job. 4th, she's very cheeky and a lot of fun in the lab, and so she's extremely positive presence, and so me and the team thank you for that. It's been really wonderful. Her work is beginning to bear fruit. She's a co-author in papers from the lab. She won the 1st and 3rd prizes for the Art and Science Prize awards in 2022, and she has a substantial first author paper that's currently under revision, and then 2 more that are coming in right behind that. And as the WeHi newsletter said, she's truly becoming Jacques Miller's thymus soulmate, was what it... So with that, Kelin, why don't you take us through your presentation? Thank you.
Thank you, Daniel. That's a surprise. Okay, so today I'm talking about how do we resolving new feature of thymic involution and regeneration. So the main character of my talk is actually the blue guy here. It's the thymic epithelium cells. We also call it TEC. And it's also the cells that I worked for 4 years. Daniel, my supervisor, told me that TEC is the most important cell in our body. That's Daniel always trying to convince people, convince everyone he met. I'm not making this conclusion yet, but I will spend a little bit time in the introduction to tell you what's their role and why they're important, even not the most. Firstly, we can see that as epithelium cells, the TEC forms this complex 3D network. And the cells in purple is spermatocytes, the developing T cells. They migrate through the TEC network, and what TEC do is supporting spermatocyte development. And there are 2 primary lymphoid organs in our body, the bone marrow and the thymus. Lots of lymphocytes are produced in bone marrow, such as B cell, macrophage, and dendritic cells, except T cells. T cells are responsible for recognizing outer pathogen, preventing cancers, and maintaining self-tolerance. So the progenitor of T cell from the bone marrow have to migrate through the blood vessels and then enter the thymus for their development. How does it happen in the thymus? Here is a schematic showing the thymic structure. The thymus has two parts, the inner medulla and the outer cortex. And the blue cell here, this one, and this one. With different morphology, they all different types of tech. They create a variety microenvironment for T cell development. The purple one is the thymocyte. Actually, 99% of the cells within the thymus would be thymocytes, but I'm using only one as demonstration. The progenitor first divide the thymus in the area between cortex and medulla. During their development, the thymocyte migration within the thymus involved three main steps. The first one, if within the cortex, the thymocyte moves towards the capsule region. Then they're migrating backwards. Finally, they reach and enter the medulla. The entire process relies on the presence of different types of tech, and I will guide you through in each step. In the first step, when the thymocyte migrates toward the capsule region, they also undergo proliferation as migration. In here, they firstly interacted with cortical tech. We also call it C-tech. There are two main things happen during this phase. First, C-tech provides notch signaling, like DLL4, to the progenitors, so that they irreversibly commit to T-cell lineage. Second, TCR rearrangement, to create various TCR for T-cell recognize different pathogens. So T-cells detecting pathogens by having their T-cell receptor, or TCR, interacting with MHC peptides on the antigen-presenting cells. So the structure, or we simply say the shape of TCR, is critical for T-cell recognizing the pathogens. The purpose of TCR rearrangement is to produce different types of TCRs. The process of TCR rearrangement is complicated, but the concept is simple. The gene encoding TCR contain a range of segments. During T-cell development, these segments are randomly combined. Each combination events different combination of different segments from different types of TCRs. So there are 10 to the power of 18 TCR created in a TCR rearrangement. Each of them bind to a distinct shaped molecules. This amazing diversity of different TCR ensure T-cells are capable for sensing any threats, including pathogens that have not existed before. But because of the process is random, many of the thymocytes make TCR is useless. Some of them, their TCR affinity to MHC molecules is too low, so they cannot bind to that MHC molecules that present peptide, like the gray one here. Some of them, they interact too strong to the cell peptide. All of these thymocytes with potential threat needs to be removed. So when the thymocyte eventually encounter the second type of tag, it's the subcapsular tag here. It's a thinner layer of cell at the thymic capsule. The thymocytes start migrating backwards. Here where the positive selection happens. The binding affinity of TCR to the MHC molecules in C-tag decided whether the cell gets survive. The thymocyte with enough affinity, which is the blue and the red here, get survive, and then they move to the next stage of development. So in short, the positive selection ensures that the future T-cell is useful. Then the positively selected thymocytes then migrated into the inner medulla regions. In the medulla, there are another type of tag, medullary tag. We also call it M-tag. M-tag has a special feature that expresses thousands of genes encoding protein normally found in one or two tissues in our body. For example, the insulin is produced by pancreas. It's now expressed by M-tag in the thymus under the control of a transcription factor called AIR. So this process is like a preview of different tissue in our body for the developing thymocytes. Negative selection is to remove the thymocytes with too strong TCR binding affinity to these MHC peptide molecules by induced apoptosis, like the red one here. This can make sure that the T cell is not dangerous to our body and is essential to prevent autoimmune disease. Only the T cells surviving all this quality control process get mature, and finally, approximately 1% of the thymocyte get mature and export from the thymus. These T cells, including conventional CD4, CD8 T cells, regulatory T cell, and unconventional T cell, they will patrol the body for any pathogens happen to be able to stimulate the TCR, provoke an immune response. I also mentioned that even the thymus has a complex structure with lots of different cell types, like the yellow cell here. They are macrophage, dendritic cell, fibroblast, and B cell. They also help in T cell development. But TAC play a critical role in supporting T cell development and are the foundation, both functionally and structurally, of the thymus. No other cell type can perform this same essential function. Even the thymus is essential for T cell development and is important for the immunity. The thymus is actually the first organ in our body undergo age-related atrophy, or we call it involution. So the thymus has a rapid growth in early age, but after puberty, the thymus starts atrophy progressively with aging. Here is a young human thymus with a decent size and clear boundary between cortex and medulla, whereas the aged thymus only tiny a little bit lower, and it's hard to tell where is the cortex and where is the medulla. The consequence of thymic involution is the reduction of T cell output as aging, and also accompanied with poor immune functions, and also increase the risk of autoimmune disease. Things can get even worse, sorry. Thymus is an immune organ which is sensitive to acute injury, such as radiotherapy, chemotherapy, and infections. Thymus can atrophy rapidly after this treatment. Graph showing here is the immune cell number before and after myeloablative hematopoietic cell transplantation. This is usually a standard treatment for blood cancer. So after the treatment, the patient immune system needs to be rebuilt. Good news is lots of immune cells start bouncing back pretty quickly, such as the neutrophil, B cells, and plasma cells. Now here's the kicker. Thymus can regenerate too and start to produce some T cell, but if you're focusing on the red line, the T cell recovery is way too slow compared with other subtypes because the thymus doesn't regenerate it as well. So the patient is susceptible to infection during this phase, and it's a major cause of morbidity and mortality. We think that it will be great. We can boost the thymus, prompt the thymic regeneration, and help the immune system reconstruction. So inside of the issue that we face with the involute thymus, we got two questions in our hands. First, what has changed during thymic involution? Second, we found that when we introduce acute injury in early life, the thymus can make a fully recovery quickly. But if we introduce acute injury in later life, especially when we have the involute thymus, the regeneration process is much slower, and sometimes the thymus may not fully bouncing back at all. So we wonder, why does the involute thymus have delayed regeneration? To answer these two questions, my talk was separated into three parts. The first part, I will show you the method I developed for visualizing and quantifying TAC in situ. The second part, to answer the first question, I will show you the apparent feature of TAC in the involute thymus, and also the role of age-associated TAC in the thymic injury and regeneration to answer the second question. And then we're coming back to the first video I just showed you. The TAC form a complex structure. We don't know where one cell starts and where one cell ends. It's difficult to locate the cells. And we're also interested in the TAC number and TAC microenvironments, the environment structure in the steady state or compared with other abnormal stage. So to simplify this, we think of focusing on quantifying and visualizing only the cellular nucleus. Here we introduce FOXN1-CRE ROSA anti-angiogenic system. You have a nuclear-localized TD tomato FOX with 2 phosphate sites, followed by a nuclear-localized EGFP. So without the CRE, other cells will express a nuclear-localized TD tomato. When we introduce the CRE, in our case, it's FOXN1-CRE. FOXN1 is a master transcription factor for the TAC development. And all the TAC and its lineage will express GFP in its nucleus. Here is a fact plot gating on the TAC population, lineage negative, EpCAM positive, and MHC class 2 positive, and expression on GFP and TD tomato. In wild type, we don't see any fluorescent protein expression, whereas without the CRE, all the TAC are TD tomato positive. And then when we introduce the CRE, all the TAC will switch to GFP expression. Here is a quantification plot. As a reminder, whenever you see this mouse with a knock-in icon in this talk, it means that we are using this genetic model. So this is what's an anti-angiogenic thymus under the confocal microscopy. The blue channel is the EGFP channel, and each of the dots representing one TAC nucleus. You can get a flavor of that. There are two populations of TAC. The first one is in the inner part of the section, with GFP cells have higher cell density, usually overlapping with the TD tomato brighter area. And then the second population has a lower cell density with a TD tomato dim area. So the first part is the cortex, is the medulla, and then it's the cortex. And you usually will have you will notice that there's a thinner layer of cells at the edge of the section. They are also quite compact. They are the subcapsular region. So in my talk, the thymus will separate into 3 parts: the medulla, the cortex, and the subcapsular. And then in the confocal, it's usually giving us the partial view of the thymus, and also in 2D. But in the light sheet, we are able to image the whole lobe and reconstruct it in 3D. The blue region really bright in the middle of the thymus is the medulla. It is so bright because the cells in this part, M tag, are tightly packed. And thus, it will show up well in the lower resolution image. And the rest of all will be cortex. So if I generate a surface for the medulla, which you already saw there, it's really clear that the thymic medulla have 2 forms. The first one, I will highlight in blue. It's a big central medulla with lots of branches connecting to each other, saying that they have a very high structural complexity. The second part is the yellow. It's a multiple small isolate that with a simple structure. They're usually at the edge of the thymus surrounding the bigger central medulla. So we now know that the thymic medulla has a complex structure that forms various microenvironments to support T cell development. Then we are now able to visualizing the tag. The next question is, how do we quantify tag in the image? We now have 2 solutions. One, in the confocal, we have high resolution image. In this image, you can count how many cells within it. But it's time consuming if you want to get a good quality image. Whereas in the light sheet image, you have relatively low resolution, but you can scan one lobe pretty fast. How do we combine together? We take the advantage of thymus has 2 lobes, and they are structurally identical. From the left lobe, we image the whole lobe using light sheet. And from this data, we get the volume of different regions. For example, the volume of medulla and volume of cortex. And for the left lobe, we cut this lobe using Vivatome to a 200 micron section and image this section in a confocal. So within this section, we can count how many cells within the section and then also the volume of different regions. So it's the cell number within a given volume. We can get the cell density. So the MTAG number would be the medullary volume from the light sheet multiplied by the MTAG cell density from the confocal. And then the next question is, where is the boundary of different regions? Here is a projection of 200 micron sections. So you will see that at the edge of the section, their thinner layer of cells is quite compact. The yellow is the surface that we generated in the analyzing software. And then if we import all this into the Immunology, you can get this. The x-axis is the shortest distance to the edge of the surface. So it's the distance to the yellow surface. And then the y-axis is how many tags in this position, also representing the cell density. So the solid line is the actual distribution, and the dashed line is the random distribution. So then you can now know that the cell distribution graphs reveal a sharp increase in cell number followed by a rapid decrease, saying that there is a cell accumulating at the edge of the section. Then this trend line turning flat, this plateau corresponds to the cortex region. To reflect this feature, we're calculating the secondary derivative. Where the secondary derivative turns to 0, that means that's the boundary making the end of the subcapsular region and the commencement of the cortex region. The value from all the sample here turns to 0 simultaneously at the precision of 25 micron. So now we know that the subcapsular region is 25 microns fit. The next question is, where is the boundary between cortex and medulla? We develop a pipeline to identify medullary region by combining tag cell density and K14 antibody staining. K14 usually labeled the thymic medulla. Here is a sample showing that tag in blue and K14 antibody staining in yellow. The green surface is the medullary surface. It's highly overlapping with the GFP positive and K14 positive area. The yellow edge is the edge of the section. So we use these 2 surfaces to segment the tag spot. For example, the spot within the green surface here is the M tag, and 25 microns to the yellow surface is the subcapsular tag, and the rest of all would be C tag. So previously in this paper, the authors obtained section from an entire thymic lobe using 10 micron fixed section. So a total of 200 to 250 sections were collected. And then they count every single section, and I asked how many tags were there. What they found is, in fact, we usually get 10 to the power of 5 M tag or C tag. But this number is actually 10 times underestimated. So there are 10 to the power of 6 M tag and C tag if we quantify in imaging. We firstly count the cells using 200 micron ribosome sections, and a total of 33 sections were collected. The number is showing in green, and we got the similar number of C tag and M tag to the paper. The blue bar is we quantifying tag using the pipeline that I just showed you by only imaging one confocal section. And we get a similar number of two imaging approach, indicating that our pipeline is highly accurate. Additionally, we were able to quantify the subcapsular tag that this population was previously cataloged as a cortical tag in FACS analysis. And there are 10 to the power of 6 subcapsular tag in one young thymus. So summarize my first part of my talk. Now we are able to visualizing tag in situ using confocal and light sheet imaging. We also be able to quantify tag in a high resolution. So you may realize that the ratio of M tag to C tag appear consistent between FACS and imaging, indicating that neither method shows a specific log of a cell type. But our method is helpful to quantifying visualizing rare population, especially like tag progenitor population. So the data I show you representing the tag in the steady state. We also be able to apply this method to the thymic dysfunctions, or we also interested in the tag dynamic during injury and regeneration. So finally, we have methods. We are able to ask our first question: what has changed during thymic involution? On the left, you can see a young anti-angiothymus. As a reminder, the purple is the medullary surface, and it has a large central medulla with highly structural complexity and then multiple small isolate medulla surrounding the big one. In a contrast, on the right-hand side, you have the 2-year-old thymus. It's an involute thymus. However, their medulla is now forming isolated lobe, highlighted in purple here, so suggesting that a partial collapse of this microenvironment. And then the most prominent feature that we see is this band light high-intensity GFP signal surrounding the thymic medulla. And then if we make a surface for this feature, it will show up later on, and then you can get that these regions compose a large volume in the thymus, and then it's unique to involute thymus. No one's seen this feature before, so we decided to study this population in a high resolution. In the young thymus, you can see a typical tag distribution with a lower cell density in the cortical region and a high cell density in the medullary region. And then in the involute thymus, except for the medulla and cortex, we have a new feature emerged that like this region. They have a really high density GFP signals, confirming that the very bright feature that you saw in the light sheet image is composed of very compact GFP positive cells. This high-density population is mostly located in the perimedullary region. It's a region around the edge of the medulla. This population is so compact that we barely see other cells, which is the TD tomato cell within this region. Remember that in our anti-angiography model, TD tomato representing all other cells within this organ. And in the case of thymus, it will be pharmacyte. So suggesting that this high-density tag from a microenvironment that do not support pharmacyte development. Because we're using the genetic model, and no model is perfect, especially when you age the animals. So the first question we ask is this population still epithelium cells? A really simple experiment you can do is staining this population with keratin. Keratin is an intermediate filament that expressed in all of the epithelium cells. Here, our high-density region are highlighted in the dashed line. They are pan-keratin positive, suggesting that this population preserve epithelium feature. But then it comes to a problem. There are hundreds of thousands of researchers before me cutting millions and billions of thymic sections and never found this guy here. And whenever a student finds something new, that's what their boss would look like. They're usually wondering, are you doing something wrong in the lab? Luckily, I found this group in US. They were also studying aging thymus, but independent from us. That's very important. They use wild-type mouse, a single-cell RNA-seq to all the stroma cells within the thymus, and they compare 2-month and 18-month-old mouse. And the young one was showing in blue, and the red one, the aged one was showing in red. What they found is epithelium cells is the only cell among all the thymic stroma that change as aging, compared with endothelium and fibroblasts. They also found a new population, they call it age-associated tag, AA tag. And these two populations only consist tag from aged thymus. Then they did an interaction map between all the stroma cells based on the expression of receptor ligands pair. In the young thymus, without AA tag, there are strong communication between different types of stroma cells. For example, strong communications of differentiating M tag to all types of fibroblasts. Whereas in the aged thymus, the emerge of AA tag 1 and AA tag 2 seems to a clear steering of interaction with age towards AA tag. It's suggesting that the AA tag co-opt the signal from other stroma cells. So except for the interrupt interaction between stroma cells, both AA tag populations show loss of the canonical marker of the epithelium subset, especially AA tag 2. They're losing EpCAM expression. EpCAM is expressed in the membrane of all the epithelium cells, where it helps the cells sticking to each other. And then the pathway enrichment analysis of AA tag 1 and 2 versus the rest of the tag population, reviewing then a downregulation of antigen processing and EMT, epithelium mesenchymal transition signature. So it's suggesting that AA tag population undergo EMT transition and thus do not support pharmacocyte development as their canonical tag population. So also they're wondering where is this AA tag population. They did a spatial transcriptomic comparing thymi from 2-month and 18-month mouse and overlapping the AA tag signature to the data set. So in a young thymus, the dashed line highlighting the medullary area, we barely see any AA tag. Whereas in the 18-month-old thymus, you will see AA tag and AA tag 2 occurring in the perimedullary region. Finally, to assess whether this AA tag as a feature of human thymus, they assess previous generated tag RNA-seq data from a 3-month-old or 30-year-old human sample. 30-year-old seems too young to us, but it actually has an involute thymus. So you and I sitting here would have a relatively smaller thymus than we were young. And this data reviewing that the AA tag only occurring in a 30-year-old thymus. So all this data suggesting that the most prominent shift in the stroma cell type during thymic involution was the emergence of unique AA tag population. So now they have the newly discovered AA tag. We have a high-density tag population that feature never saw before. What's the relationship between them? I don't know yet, but now Daniel's face has smiled. He started to believe that my discovery is new. So in previous data, by staining pancake, we confirming that high-density tags are still epithelium cells. I continuously staining different subunits of keratins and compare them with the RNA-seq data. So here, all of the AA tag 1 and some of the AA tag 2 express K8. And we see the K8 staining in the high-density region. And some of the AA tag, just a little bit AA tag 2, were expressed K5. And the K5 staining also occurring in the high-density tag region. AA both AA-TAC1 and AA-TAC2, they do not express K14. We don't see any K14 staining in the high-density region. This is interesting, because K5 and K14 usually co-express in MTAC, but now we are missing K14 expression. But coming back, by combining this subunit expression, we now know that the age-associated tag is high-density tag. So now we are becoming collaborator and continually study this population. The data I just showed you is just part of the story, and it's now the paper is under review. If you're interested in, it's also in bioRxiv. Please scan the QR code. The summary of this paper for the PhD student in this here. Never try to convince your boss by yourself. Find someone else. Okay, the story needs to continue. In imaging, we can easily locate this population, but we also lack of method to purifying this population. So in previous RNA-seq data, we know that this population loses lots of canonical marker. So we wonder how does it look? We used our anti-NG model, gating on the lineage negative and GAP positive population, which is all the tag. In the young anti-NG thymus, all the tag are EpCAM positive. Whereas in the 12-month-old anti-NG thymus, except for the EpCAM positive population, you get an EpCAM negative, GAP positive population. So this is correlated with the AA-TAC2 that they're losing EpCAM expression. And then we are analyzing the EpCAM population first. In the young thymus, you either get Li51 single positive C-tag or UEA1 single positive M-tag. But in the aged thymus, except for the C-tag and M-tag, you got this Li51 and UEA1 double negative tag. And then we're also analyzing the EpCAM negative population. It seems like this EpCAM negative population consists of Li51 positive and double negative population. So we conclude that the AA-TAC1 is the EpCAM positive, UEA1 and Li51's double negative population. And the AA-TAC2 is the EpCAM negative tag. Then we wonder, what's the role of AA-TAC in thymic hemostasis? The first one is the FOXM1. Like I said, it's the master regulator of TAC. It plays a critical role in thymus development, function, maintenance, involution, and even regeneration. So we wonder the expression level of FOXM1 in AA-TAC population. But firstly, as you will see, if we're getting on the GAP positive population, as aging, the FOXM1 expression decrease. And then we assess the expression level in different population in the aged thymus. MTAC high is the MTAC express high level of MHC class II molecule. It's an active population within the thymus. So the MTAC high will have a higher expression, high level of FOXM1 compared with MTAC low and CTAC. We're also seeing that both AA-TAC1 and AA-TAC2, they barely express FOXM1. So in our model, remember anti-NG mouse, all the GAP positive cells are once express FOXM1, but now we know that AA-TAC no longer express FOXM1. And then we're wondering, how do AA-TAC progress with age? Does it occur at a certain age and maintain? Here I quantified the live sheet data from 8-week, 1-year, and 2-year-old thymus. As aging, the thymus start atrophy, also the medulla. In a young thymus, we don't see any AA-TAC population, but this population increase as aging, suggesting that this population expand. If they are expand, how fast do this AA-TAC population proliferate? We did a simple experiment by injecting the EDU to the mouse for 3 days. EDU is a thymidine analog that's incorporated into DNA during S phase of the cell cycle when DNA synthesis occurs. So the cells, once proliferate, will be labeled by EDU. So like I said, MTAC high is an active population within the thymus. So by the end of day 3, there will be nearly 50% of the EDU were labeled by 50% MTAC high labeled by EDU. But we don't see lots of a label of MTAC low and CTAC. We also, in AA-TAC1 and AA-TAC2, we don't see lots of cells were labeled by EDU, suggesting that this population has a slow turnover rate. Finally, if the AA tag is not highly proliferate, which means that they are maintained in a certain state, and then we ask, what do they depend on for survival? We assess the expression level of BCL2 family post-survival protein. In a young mouse, the highest level of BCL-XL and MCL1 were found in the MTAG high population, and the BCL2 were in CTAG. BCL2 level were in CTAG. In the aged mouse, we see a similar expression pattern of MTAG high, MTAG low, and CTAG. But in the AA tag 1, it has lower expression across BCL2, BCL-XL, and MCL1, suggesting that this AA tag population is sensitive to cell death. But like I said, they have a slow turnover rate. What we know about this population right now? We know that they do not support pharmaceutical development according to the image and single-cell RNA-seq data. We know that this population accumulating as aging, but they lose FOXM1 expression. They also have slow turnover rate. What do they do? The true answer is this question mark here. We don't know yet, because we can't generate enough data in the steady state. So we decided to challenge this population. And leads to my final part of my talk, that we're asking the second question: why does involute thymus has delayed regeneration? Previously, our collaborator, they did a sublethal irradiation to the young and aged mouse. So after the irradiation, the thymus start atrophy, and then they would start generated. By the end of day 28, the young thymus get fully regenerated. Whereas in the aged thymus, in day 28, the thymus still not fully regenerated, and this situation continued to day 42. We simply repeat this experiment using our NT-NG model. And we harvest thymus at day 28. We ask what's the feature of thymic involution impaired thymic regeneration. Edwin, my committee chair, he always criticized this part because I previously showed the data by creating an animation. Edwin, this is the real data. This is the raw data. Just for you. Are you happy? But I will still make the animation for the rest of the audience. So on the left-hand side is a 1-year-old NT-NG thymus. The medulla highlighted in purple here, you see that they start losing structural complexity. And we also have a certain amount of AA tag highlighted in blue. On the right-hand side is the thymus 28 days after the irradiation. The first thing you will see later on is the thymic medulla now only the small lobe-like medulla left over, and no connection between each other. And the other striking feature is the AA tag population taking up more space within the thymus. I quantified this image, and after the irradiation, the size of the lobe and the medulla and cortex were decreased, but the volume of AA tag population increased. Also quantifying the tag number by using the method I just presented to you guys. We confirmed that the AA tag number also increased. So in all, we know that this AA tag population expand after acute injury. So then we come back to the RNA-seq data. At the day 7 after irradiation, in a young mouse, a broad upregulation of endogenous epithelium regenerative factors were found, such as FGF and BMP. But in the aged thymus, our interaction analysis suggesting that the regenerating interaction of BMP and FGF pathway are skewed towards the AA tag. So if you compare this to this. So suggesting that AA tag co-opt the tonic signal from the tag population at the basal line and then reparatively cues after damage. Summary of my second and third part. We now found a tag population that emerged with aging. These age-associated AA tag create an aberrant thymic microenvironment that do not support thymocyte development. This AA tag population can expand after thymic injury, so it's correlating with an impaired regeneration. So we think that we have some hypotheses. We think that as thymus aging, one of the population of tag, they're losing their expression of FOXM1. So this population transitioned to the AA tag. In the young thymus, after the injury, IOC fibroblasts and endothelium cells would express the regenerative factor like IL-22, KGF, that support regeneration, and then would subsequently supporting the thymic regeneration, alpha-amopoiesis. Whereas in the aged thymus, this regenerative factor was still by the AA10 population. However, like I said, AA10 does not support pharmacocyte development, so that we have a delayed regeneration. To test this hypothesis, we have some experiment that coming on. So first one is we have use the Loxco mouse. Sarah, a few weeks ago, introduced you guys this mouse model. And we give this mouse a drug, and then all the cells will label by a genetic barcode, and then we just living in the shell for a year. And after a year, when we're tracking back this barcode, we would know where this AA tag are arising from. The second one is we are doing a spatial transcriptomic analysis, which is MERFISH, by comparing the young and age, and day 7 after the irradiation and day 28 after irradiation. Day 7 is the time point that the thymus start regenerating, whereas the day 28 is the thymus still not fully regenerating. By comparing this data, we're able to ask what causes delayed regeneration in the immunothymus. So finally, I hope that I can convince you guys that the tag is the most important cells in our body. So a little bit relaxed time just for me. I'm really lucky to join this lab, but as a PhD student joining in 2019, we know that we already have COVID-19, so we have like this happily playing mini golf and transition to this everyone sad face sitting in front of computer. And just clarifying that Daniel's sad face is not because of my data, it's because of COVID. But finally our lockdowns end, and then now we have lots of lab meeting or lab lunch. I also thank the friends that in China or in Australia. Sometimes because of COVID, you just have to PS yourself into their photos. But I'm very happy that I made new friends in here, Weihai, or overseas, and sometimes even underwater. Finally, I thank my collaborator to saving my life in the lab. Finally, thanks all the present, all the members in Grey Lab, past and present. Daniel, thank you for inviting me into the lab. Julie, without you setting up all the mouse model, this project won't happen. Rima, welcome back to Weihai. And I still remember the funny thing that we did is we're sitting in front of the camera and you're telling me how to do a thymic prep. And Lucy, Karis, thank you for so much support and then helping me to giving lots of advice for this talk. Make sure you're always my buddy in the lab. Michael, Asma, thanks, welcome to joining Grey Lab. And thanks to my committee, Edwin, Jane, and Dell. You give me so much support during these 4 years, and everyone in the imaging facility. Because when I firstly learned how to use microscope, I was too scared to touch anything. And I would just rush to the office and say, please help. Thanks, Marina, as my co-supervisor. You're introducing me lots of imaging techniques. Loki, thanks for provide lots of pipeline to analyze this image. Pradeep, you generated lots of like the surface you generated to help me analyze this nicely image. Sabrina, you were always my perfusion buddy. But everyone in biosurface, especially Laura, Kate, Zach, Haley, taking care of my lovely naughty boy. And also lots of people at Weihai, especially Caleb, Stella, Charles, Bridget, and also Kylie, you are the best imaging body in the imaging. Oh no, in the immunology, sorry. Yes, family and friends, especially my parents, because they don't know what I'm doing right now. They don't know what I'm doing right now, like lots of PhD students' parents. But, well, at least after 4 years, they know thymus is an organ in our body. Especially not thyroid. Thank you, Mom. Great progress. Okay, I'm happy to take any questions.
Okay, thank you very much, Kelin. We've got plenty of time for questions, and if you're joining us online, please put them through on the side note and I'll read them out. We've already got a comment from Edwin. He says he's very happy with the raw data, Kelin. Thank you very much. You can see what I meant about Kelin supervising others, right? So, but maybe we'll take a question from the floor. I'll give this to Tim.
Just wondering, did you look at any senescence markers during this aging or radiation experiments?
We are trying to do this in the murfish, and then yeah, so it's ongoing. But we haven't right now. Sorry.
Thank you. Then there was one right up there. She was first.
Congratulations on an awesome project and a great talk. That was incredible. Well done. I was just wondering, with your AA techs,
Yep.
Do you think that they are their own sort of population, or do you think they are MTech hides that have just lost the ability to present MHC?
From our single cell data, we actually think that this is a population of MTech1. It's MTech1 that losing its FOXM1 expression that causing the AA tech. But this is single cell RNA-seq data, so we don't know, like we can't know for sure. Yeah, but it's definitely from a population of tech that it's occurring like in the young thymus.
Yeah, right. Cool. Thank you.
I think that question was about whether they're just like MTech that have just lost MHC rather than them wherever they come from.
Okay.
So do you have any?
Well, but it's not definitely not like MTech, but because they don't help. Like MTech supporting negative selection, but structurally there's no cell within this region, so they're not doing the same job as MTech. Yeah.
Reiterate a fantastic talk. In the aged mice, the lobes seem to be what sticks around, and the bridges between the lobes and the younger mice is what disappear. So in your 2-month-old mice, is there anything special about what's in those epithelial bridges between the lobes?
What do you mean epithelial bridge?
So it seems like there's a branching structure at 2 months, and you've got lobes and then bridges and then lobes. You've got like these large densities of cortex. And then in age, you've got isolated lobes. Is there any structural or any phenotypic variances that you've seen in the AA-tex between, sorry, the M-tex between the lobes?
Just a hypothesis here. So we think that different lobe representing different cell peptide antigens. So it's a different like microenvironment. So actually, there is a paper long ago, they using 2D structure. So in 2D, you can imagine the lobe is also separated. So it's saying that these different lobes in the young thymus, they are actually from different progenitors. So suggesting that they actually different. Yeah, so then we think that the sum of the lobes was becoming AA tag in the aged thymus, so they're not connecting to each other anymore.
Maybe speculate a little further about what the cues are that guide the thymic medulla and potentially the identity technique. What an idiot. I turned it off. Did you want to expand a little bit more and discuss those cues that guide where the thymic medulla goes and therefore those AA-tech ribbons? I'm talking about the vasculature here, right? And then what cues might also induce the AA-techs?
So, Daniel is asking what could introduce the AA-tech populations. And then we think, so the first hypothesis is from me, not from Daniel. It's hard to prove, but we think that it's revolutionary preserve that populations, constantly there's a population of MTech, they would becoming AA-tech, because we know that in the aged thymus, some of the antigen present, some of the antigens self-peptide will no longer present in our thymus. So this population will becoming AA-tech. But what's that? Is that a correct answer?
That'll do, but there's also a lot of adipose tissue that comes into the aging thymus through and it's associated with the blood vessels. So that might be what Ryan's referring to, that collapse between the bridges. It might be defining those cues. Oh, question here. Pass it.
Thanks, wonderful images and rendering. I had a question about the clearing protocol. So you use PACT, right?
Yeah.
The passive one. So in terms of your volume measurements, how much of the swelling that is called, like, so if you soak it over time, there is some natural swelling that happens with the tissue.
Yeah, correct.
How much do you think that would impact your volume assessments, like in terms of a true representation of the natural tissue state?
So it does swelling. So if we do the quantification, we always treated the section and the lobe using the same methods. So that would be 1:1. There's no problem for the quantification. But we also did a experiment that we just staining the thymus with iodine. So that increase the contrast of the medulla and cortex. So the structure in here is pretty similar. So we think that the swelling does not affect much of the structure.
Okay. And then the detergent would clear away any fat and lipids and stuff. So if adipose tissue is indeed important to those bridging connections, how much would the clearing of lipids impact those structures?
So it's the fat, sorry?
Yeah, if adipose tissue plays a big role and you're clearing away any lipids with the PACT protocol, how big of an influence would the clearing have on structures that you might not be seeing?
The true answer is I don't know. But our model, because it's based on the NTNG, so structurally, we're only focusing on the GFP channel, so it is what it is. Yeah.
Yeah, we can maybe avoid clearing or look at some markers for adipocytes to try and get around. So excellent point. We have an online question here from Dale. Yes, congratulations, he says, Dale Godfrey. Can you speculate on the physiological role for AA tech? And he asks whether they make the cytokines like IL-15.
They don't make much of cytokine. So that's all I can say. It seems like a really silenced population. They have low expression BCL2 protein. They don't express MHC class 2. So no.
Yeah, I have a question that goes in a similar direction. Do they express anything that is like seen in other populations of aging cell types?
Of other aging population,
because like a lot of cell types do have like weird different gene expression profiles when they age.
Yes, we trying to stain some of the keratin. Keratin 10 is usually expressed at the end stage of the population, using of the aging, but it's not. So just for us, it's just really weird population, I said. Yes, it doesn't express lots of things, but it does express some of the progenitor markers. We actually trying to use these progenitor markers, coding-free, if you're familiar with TET, to isolate this population.
Rima.
Congratulations, I'm so proud of you. I might have missed this, but in your aging model, where you looked at AA techs that were radiated, sublethal radiation, I think?
Yeah.
Yeah, so you see an increase in AA techs, right? Is it because of proliferation or is it because of transition from traditional techs, say M-tech or C-tech, into AA techs? Did you look at EDU or BRDU?
We haven't looked at the EDU yet. I was going to answer your question, but what's that? It's a transition from other tech. So firstly, in the young thymus, if you irradiate the thymus, we don't see this AA tech population. So at least it's not from a healthy progenitor, a healthy population of tech. So that's why we coming, but it has lots of receptor of the FGF and BMP4. So we think that this is because they are stealing this factor, and then they themselves proliferate.
Thank you.
Beautiful talk. One of the things I was thinking when I saw those clusters was the absence of separator cells that then just kind of brought them together and they were a bit sticky and whatever. I wondered, because I know there's thymic reaggregation techniques, I can't remember exactly what they are, but can you mix young and aged and see if they disappear?
We are trying to do that, but as I'm writing my thesis, not happening right now. Yeah, but we, for the reaggregate, we do trying to castrate the mice, which the thymus can regenerate, but they're still there. So it's really seems like really hard to get rid of them.
Okay, we've got 1 minute for any last burning questions, but I think we put Kelin through the ringer enough. There's no more online, so it just remains for us to get them out and bang them together for Kelin. Excellent job.